A very good morning to all the students and my dear colleagues. A warm welcome to today's online guest lecture on a practical guide for designing animal experiments in biomedical sciences. So on this regard today we have a special guest Dr. Lakshman Kumar Puli, who is working as an assistant professor pharmacology and toxicology at School of Pharmacy and Technology Management SVKM NMIMS University Hyderabad. Before starting this session, let us have a brief regard our today's speaker. Dr. Lakshman Kumar Puli have completed his Bachelor of Pharmacy, Pharmaceutical Sciences in Kakatiya University and his Master's in the specialization of Pharmacology and Neuroscience in Rajiv Gandhi University of Health Sciences from 2003 to 2005, and his Doctor of Philosophy at University of Eastern Finland on the aspect of neurodegenerative disease, Alzheimer's disease, neuropharmacology and neuroimmunology from 2006 to 12. And he is also worked at the AI Vachanian Institute of Molecular Sciences for 6 years and postdoctoral fellow and Leon Levy Neuroscience Fellow at Weill Cornell Medical College, New York City, and postdoctoral researcher at Karolinska Institute at Sweden, and worked as assistant professor at NIPER Raebareli, and also he is currently working as assistant professor at SVKM NMIMS in Hyderabad campus. He is a very well experienced biomedical professional with more than 10 years of exposure to brain research and institutes in Finland, USA, and Sweden. Along the way, he gained experience in, as well as expertise in many skills regarding the pharmaceutical drug discovery and development. It is very glad to have you, sir, today. And from the Department of Pharmacology, Chalapati Institute of Pharmaceutical Sciences, we welcome you for this online session on a practical guide for designing animal experiments in biomedical sciences today. Hope this all goes well. We welcome you once again, sir. Thank you. You can carry
on. Thank you, Dr. Ishwar. Am I audible?
Yes, sir. You are audible.
Yeah. Very good morning to all of you. Thank you, Dr. Ishwar, for that kind introduction. I also thank Professor Narendra Babu, who is the HOD, for extending the invitation for this guest lecture. And it is indeed a great opportunity for me to share whatever knowledge I have with your students and with your faculty members. And these are very testing times for us. And COVID lockdown time, basically we have to upgrade ourselves in terms of knowledge. We have to sharpen our tools. As a pharmacologist, also we have to keep an eye around, because so many people are talking about so many things which we know that as a pharmacologist, which is not true. So today, the topic that I have chosen is very basic one. With the after conversation with Dr. Ishwar, that the topic should be useful for the PG students, MPharm students, and as well as for UG students. So I thought that this very basic topic would make sense, and that's what I'm going to explain. So if you could make me co-host, I would be able to share my slides. Yeah, thank you.
Yeah, yes, sir. You can proceed.
Yeah, I'm sharing my screen. Is my screen visible?
Yes, sir, it is visible.
Okay. All right. So, as Mr. Ishwar said, I started off with the School of Pharmacy and Technology Management, NMIMS University in Hyderabad last year. And this is a brand new college established in Jachalla. We have a huge campus, and we have set up School of Pharmacy and Technology Management. Alongside, we have School of Business Management, School of Science, School of Law, and also School of Commerce. And so we're just setting up, and we're glad that we have well-established laboratories, and I'm thankful for our management. At the same time, today, the topic, as you could see, is a practical guide for designing animal experimentations in biomedical sciences, not just restricted to pharmaceutical sciences. So what we are going to, so this is the first slide we have. I think most of you might already recognize the person, Paul Ehrlich. You all know him for the concepts of immunity, right? Receptor ligand binding studies. He is a well pioneer in it. So that study was done somewhere in 1908, and from there the concept of antibodies took place, and then for those antibodies to be used as a diagnostic purpose, it almost took 38 years. Way back in 1940, a guy by called Coons, he tagged these antibodies with some peroxidases, and he tried to use it for identifying structures or proteins inside the cells. So you just see how many years it took for a simple incremental advancement in the science. And again, from 1940, by the time this particular technique was applied to the neuroscience, it took again another 20 years. So the first brain immunohistochemistry studies, they appeared in 1970s. So the point in this slide is that research is incremental. Don't expect that if you spend 1 lakh rupees this year, by next year you will have a research paper and then that will make an impact in the society. No. If someone is saying like that, then probably he's a politician yeah, he might be a politician. Only politicians say so. True pharmacologists know that just within 1 year or just within a matter of 2 years, it doesn't happen. Even if this is the same case with COVID vaccine. I mean, they're saying that within 1 month they're going to come up with a vaccine. So that 1 month is yet to come. Let us hope so. We will soon have a vaccine. But then again, the point I'm going to make is that research is incremental. Koncham koncham advances would take place, and these advances, by the time we realize its true potential, it would be already decades. So what I want, let us move on, and what I would be covering in this lecture is I would be explaining you certain critical points like how could you design an animal study, and before we actually going there, we'll see is it really necessary to do an animal study, and what could be the along the way we would also touch upon the various limitations as well as challenges that one would face, and I'm damn sure most of the MPharm students and PhD students already know about this, and I would try to explain these steps or these concepts in designing the animal studies using two of my own in vivo experiments I have done. Okay, so that's for 40 minutes. I think this is a very small objective for today's lecture. And I mean, if you are not clear of anything, please stop me at any point of time and do ask. And I would prefer it to have in an interactive way. I mean, we can always have question and answers at the end as well. But then if anything is not clear, just let me know. Okay, so those were the objectives. Moving on to why do we really need animal experiments? I mean, this is such a basic fundamental question. And as a pharmacologist, as a pharmacy postgraduate, you all know that we study about complex disease mechanisms, we study about drugs, we study about the biomarkers, we try to see if a particular drug is going to be useful or not useful. We also at the same time try to find out whether it is safe or not. But then again, all these questions can be answered in human beings as well. But then who's going to be ready to take that first dose? No one is ready. So but then again, animal experiments really help us in understanding or unrevealing to us the various disease mechanisms which is not possible to carry out in humans. At the same time, the animal experiments can also tell us or identify the drug targets. Nowadays, people are talking about the biomarkers. Why are biomarkers important for us? For example, the Alzheimer's disease, which is very near to me and dear to me. I mean, I have worked with this for the past 10 years. Biomarkers plays a very important role. Why? Because by the time the patient gets the symptoms, he is in his 70s. Elderly people get this Alzheimer's disease. Alzheimer's disease is a disease wherein which a person loses progressively his memory functions and his executive functions, and his brain gets filled up with a toxic protein. Okay, so by the time the patient develops the symptoms, it's almost 20 to 30 years past the first phase. So, for example, in 70 years or at 70 years of age, if a person is developing a memory loss symptom, and but actually the disease process has begun already when in his 40s itself. And what will tell me whether the disease process has started or not? It is the biomarkers. So it is very much important that we look at these biomarkers. Okay. So biomarkers are very, very, very important. At the same time, as a pharmacologist, one of the fundamental things that we do is we always try to compare whether the particular treatment is going to work or not. We need to have that kind of a predictive ability, and that kind of a predictive ability. What would we do with that? It gives us confidence. So those for those experiments also we need animals, and more than anything. we need animal experiments to make sure that the drugs or the biomarkers or the tests or even the formulations, whatever we are using, that are we are applying to the human body, they are non-toxic and they are safe. And that is only possible only if we use if we carry out properly these animal experiments. So yes, definitely our society needs animal experiments. There is no doubt about it. But then again, we can the conversation could go around what type of experiments might need animals and what type of experiments animals may be replaced. That conversation will go on for a long time. Then as I said, I mean, okay, animal experiments are important, but then again, are they of any use? What is their use? I mean, what is the use of the data that we produce? So one of the main what do you call advantages of an animal experiments is that we can actually model a human disease in an animal. Now what advantage do we have? So we can, so if we know, if we can model, for example, let's take the case of Alzheimer's disease. If we can model the symptoms that we see in human disease, the same kind of symptoms if we see in animals, then that would allow us to test out treatments, that would allow us to find out new biomarkers, that would allow us to predict the outcome of a particular drug candidate even before it is introduced into human being. That would also allow us to predict the toxicity also. So one of the main uses of animal experiments is it allows us to optimize disease models, animal disease models or human disease models. And again, when you talk about disease models, there could be plenty of validities you can carry out. It could be construct validity or face validity or predictive validity. Construct validity means the way the disease is constructed in the human, the same construct is also present in the animals. So if that is present, that is called as a construct validity. However, there is also something called as a face validity. Face validity means the way the disease shows up in humans. For example, Alzheimer's disease has got these protein deposits inside the brain. So my animal model also should have those protein deposits inside these animals. Then only it will have face validity. And then predictive validity. For example, in my animals, if I'm giving a particular antibody and removing all the toxic proteins from the brain, will it be predicting the same effect in humans? So that validity is also important. So animal experimentation allows us to develop and optimize human disease models, which would, I mean, that would save lots of money also for pharmaceutical companies. Because if they already know that this particular drug candidate is not going to work, they're not going to spend the resources nor the money. At the same time, at the same time, every what you call disease has got a pathomechanism. You all have studied pathology. So if you know the intricate steps in the pathomechanisms, then you can delete one mechanism or delete one key molecular player and validate that mechanism. And once that mechanism is validated, you can choose any part of that entire pathway as a target. So again, the animal experiments will be very, very useful for validating a in vivo pathomechanism. At the same time, for example, there is a anti-inflammatory properties there. So again and again, you're messing up with your drug molecule, changing the drug molecule. I mean, chemists do it very nicely, but then every time they change it, you have to make sure that it is not losing its biological properties. For example, I'm interested in a biological properties that is called as a lowering of blood pressure. That is the property I'm looking at. So every time I'm changing the drug, I have to make sure that that property of lowering the blood pressure is maintained in that molecule, and that again that I can do it using an animal experimentation. So what does animal experiments give us? Animal experiments give us the data. This data gives us the confidence that yes, this is the drug molecule, yes, this is the disease mechanism, yes, this is the target, which I can spend the rest of my resources and my money. I can bet my money so that this can be successfully translated for human use. So that's why those are the uses of the animal experimentation. Then again, the question comes: do we really require animal experimentation? Yes or no? Some people would say yes, and some people would say no. And I think the answer to this question would be depending upon the context. Okay, depending upon the experiment. So there are certain experiments for which animals can be totally replaced, but there are also certain experiments wherein which you definitely need you definitely need animals. For example, these toxicity studies, and not only the toxicity, the efficacy studies, preliminary efficacy data. You need to get it from the animals. So there is no yes or no kind of an answer for this. The answer for this lies with in the context of the question that you're asking. So, going to the next slide. So, how do you design a proper animal experiment? So, this is the entire workflow is there for designing an animal experiment. It all starts with what is called as a knowledge gap. You have to identify a knowledge gap, and then that knowledge gap. Of course, there are plenty of knowledge gap. What do you mean by knowledge gap? We'll see. Then once you identify the knowledge gap, then you have to decide whether there is any need to fill that knowledge gap. Knowledge means what? Knowing something, right? So what is causing Alzheimer's disease? Nobody knows. No one knows, and that's the knowledge gap. And is there any need to fill that gap? For some questions, yes. For some questions, no. For example, I mean, my blood is red in color. Why is it not green in color? You can do a experiment. You can feel that knowledge. But then again, is there any need? I don't think so. I mean, I'm happy. I'm glad that my blood is red in color and it is doing its function fine. But then why should I be bothered about why my blood is not in green color? So what I want to say is that whenever you find a knowledge gap, you already have to look at, is there any need to fill that knowledge, right? Is there a need to generate that knowledge? So once you find that there is a need for generating the knowledge, you create a research question, like research. Can I find a cure for Alzheimer's disease? Can I find a cure for COVID-19? A research question. These research questions could be very global questions, very non-specific, or they could be very specific questions like, is my vaccine going to alleviate all the symptoms of COVID-19? That's a very valid research question. Still, it's a very broad question, but then it can be narrowed down. We'll see how. Once we have a research question, the next step is hypothesis. So from the statement of the research question, you create hypothesis. Hypothesis is nothing but a statement for which there is no evidence or data, for which you will carry out the experiment and derive the data. Okay, so hypothesis is the conclusion that you would actually derive after carrying out an experiment. So you make hypothesis a priori before you carry out the experiment. Okay, my hypothesis is that this particular antibody is going to remove the brain amyloids, or this particular antibody is going to kill all the SARS-CoV-2 viruses. That's an hypothesis. There is no data to support it. But then again, in order to make this hypothesis a valid one, you're going to test it by carrying out an experiment, an animal experiment. Okay, in this regard, we're talking about animal experiments. Experiments is only animal experiments. So we are going to test it in an animal model. And for that, first you have to choose what kind of animal model you want to use, because there are plenty of animal models. And again, no model is a perfect 100%. You just have to decide what is that you want to measure. And then based upon that, you will choose the model. Okay. Once the model is ready, once the research question is ready, once the hypothesis is ready, then you have to frame your objectives. Objectives are these statements, the straightforward, simple statements that really tell us in one sentence what you are up to in this particular experiment. For example, to evaluate the total number of neurons in my brain. That's one objective of my experiment, if I really want to do that. Okay, to identify or to test the memory or the cognitive levels of these particular mice. That's one objective, behavioral objective. To quantify the amount of amyloid present in the brain of a mouse. That's another objective. So objectives are very, very specific. Remember, research questions here, they need not be very specific. They can be global as well as then they can be filtered down to a specific one. Whereas objectives needs to be very specific. It needs to specify the most of the details of the experiment that you're going to do. Okay, once you have the objectives and ready, then you can go on and carry out an experiment and derive the data. And once the data is done, then you generate the report or generate the knowledge. And that knowledge can be actually, you have to find out whether this knowledge is filling the gap or not. So there could be only two outcomes. So you would either fill the knowledge gap or you will not fill the knowledge gap. If you fill the knowledge gap, well and good. And then if you don't fill the knowledge gap, then you ask yourself what went wrong or what was the situation. What probably is it the right way to do it, or there was something else you are missing. So this cycle would continue. So For any experiment, you have to start with a knowledge gap. You have to convince yourself that that knowledge needs to be generated, so that there is a need for that particular study. Then when you find out a particular need for that study, you have to find out whether that research question can be answered or not, frame that research question. And once you have the research question ready, then you can frame a hypothesis, for which you know that this is going to work, but then there is no evidence for it. That's the hypothesis. And then you choose the animal model and make to clarify your objectives, carry out an experiment, generate the data, and then you report. So this workflow carries on for each of those experiments you're going to do. So let us try to find out what is the knowledge gap. We already touched upon it. How do you know that there is a knowledge gap? For example, we know that Alzheimer's disease is a disease which robs people's memory. What we do not know is what is causing the memory loss. So there is a continuum of knowledge, and then that continuum of knowledge, there is certain breaks are there, right? So I know that Alzheimer's disease is because the brain loses lots of neurons. The brain, the lots of toxic proteins inside the brain. But I do not know whether the loss of memory is because of the protein or not, toxic protein or not. So that's the gap I have. So I have to fill that gap. So that gap I'm able to tell only if I do a proper literature research. Okay, you might have heard the word go to the library, do literature survey. What you're trying to find out? You're trying to find out the missing dots. So all you have to do is, I mean, it can be done by literature survey, or these days you could actually read a review article. I mean, review article is a very perfect way of finding the knowledge gaps. Also, you can also look about the interviews of popular scientists. These scientists, in their sentences or in the way they express their science, they already give up certain hints to find the knowledge gaps. And also do read the press releases of the pharmaceutical companies. Pharmaceutical companies never share the knowledge gaps that are going to fill up. But then if you really look at the press releases, you would be able to identify. I mean, you have to get the knack of, you don't have to focus on what they're telling, you have to focus on what they're not telling. So that way, you can already identify what these companies are up to. And if you know what these companies are up to, you can really tell what knowledge gap they're going to fill up. So basically, knowledge gap is there, and you have to identify the knowledge gap, and your literature survey and review articles and your one-to-one interviews with your guides or any consultant would tell you what sort of knowledge gap is a relevant particular disease or not. That would be answered. So once you have identified the knowledge gap, then you have to convince yourself and your guide that, okay, there is a knowledge gap, but then there is also a need to fill that gap. For example, these needs could be different types of needs. There could be a societal need. What is the societal need? For example, Alzheimer's disease is a disease of elderly, right? And the elderly cannot take care of them. So hence you have to spend, governments have to spend a lot of money to take care of them. So that is a monetary burden on the government. So even for the society also. So there is a societal need to find a cure for that particular disease. So there has to be a societal need. And then again, it's not necessary that there is a need in the society would always be commercially viable. For example, I'm working on an antibody, right? And that antibody can be sold as a cure for Alzheimer's disease. So that's very commercially viable. So that should be able to make money. Okay, for example, my research comes up with a new biomarker. Biomarkers are the hot topics nowadays. Why? Because they have a very huge commercial value, because they could be developed into a diagnostic kits, right? So whenever there has to be a commercial need, there has to be a societal need. There is also something called as academic need. For example, let's take the same example with Alzheimer's disease. Lots of neurons are dying. So academically in my lab, what I can do is I can actually count the number of neurons in the mice or a human brain, a post-mortem human brain. But then again, will anyone give me money if I count the number of neurons in a mouse brain? No. So even though it is not interesting from a commercial point of view or from a societal point of view, from an academic point of view, it is very much important for me to count the number of neurons or the number of microglia inside my brain, because that already tells me something about the disease mechanism. So that is my academic need. And then there is also the need for the pharma companies. So pharma companies, they have their own issues to deal with. Okay, pharma companies, for example, they want a quick assay. They have a big biomarker study going on, and they have to develop an assay with which they can, for example, take the case of COVID-19, right? Most of the pharma companies, they are working on the vaccines. But then, is your vaccine successful, or how would you know? You need to develop an assay. So these pharma companies also have certain knowledge gaps. They identify the knowledge gaps in the research projects, and there is a need for those knowledge gaps to be filled. So just having a knowledge gap is not essential, but also there should be a need from the society. There could be a commercial need, there could be academic need, there could be a need in the pharma companies. You just have to identify that need so that the need of the study is properly demonstrated. And once the need has been identified, the next step is you have to frame your research question. Okay, let's take the same example of Alzheimer's disease and antibodies. So my research question is, can immunotherapy or can antibodies or can passive antibodies reduce the brain amyloid in Alzheimer's disease? So this is the research question that I have framed because I'm working with antibodies, because I'm working with Alzheimer's disease. I want to ask the question, research question, can antibodies reduce brain amyloid, which I assume to be present in the brain, which I assume to be toxic in the brains of Alzheimer's patients? Can I remove or can I use this particular strategy to treat Alzheimer's disease? So that's one research question. But then mind you, the research questions are not a single question. Typically, the research question can be framed into several questions. So the topmost question would be a very broad question, like, can I cure Alzheimer's disease? Very broad. Can I cure Alzheimer's disease with antibodies? Can I cure Alzheimer's disease with antibodies by reducing their brain amyloid load? Can I quantify the brain amyloid load inside the brain? Can I? So as you see, as we are coming down, you are actually narrowing down all the questions from a very broad global research question. You're coming down to a very specific research question here, which is how much of the percent of hippocampus is covered with amyloid in my brain sections. That is, I'm very much specifying the brain region. I'm very much specifying what I'm measuring. I'm measuring amyloid areas. Okay, I'm very much specifying which organ, that is the mouse brain sections. So while framing your research question, you frame a set of questions in such a way that the topmost question is a very global question, not specific one. And then the lowermost question is a very specific question. So when you generate the data, you simply answer the question like, yes, 30% of the hippocampus is covered by the amyloid, and then 30% of it is reduced. Okay. So basically you can, yes, you can quantify the amyloid positive areas in the brain. Yes, I can measure amyloid pathology in mouse brain. Yes, I can reduce the brain amyloid to 30% in Alzheimer's disease. So this way, if you frame your research questions, break them down into little small parts, write them down. Once you get the data, that data will actually answer these research questions. But then again, you have to be very, very critical. You have to sit with your guide and you have to frame your research questions in such a way that you would be able to answer these questions. So once you have your research question ready, what's the next step? You have to prepare your hypothesis. As I said, that hypothesis is nothing but a statement which is derived from your research question. My research question is, can I reduce the brain amyloid by 30%? Hypothesis would be yes. But then again, hypothesis is the statement is based upon the research question, but then again, there is no basis or there is no scientific evidence for it. You are actually going to do that experiment and provide support to this hypothesis as a result of the data that you produce. Either you will accept this hypothesis or reject this hypothesis. Okay, so this hypothesis creation is quite easy. So if you have your research question ready, the hypothesis will be, yeah, yes, my antibody is going to reduce the brain amyloid. That's my hypothesis. But then what would be my null hypothesis? Null hypothesis is more of a statistical application. It says, no, my antibody is not going to reduce the brain amyloid. And if at all the data disagrees with me, then it has to reduce the brain amyloid, and then I will reject the null hypothesis and I will accept this hypothesis, which is based upon the research question. Again, while question about while making the hypothesis is that this hypothesis needs to be really practically possible and it should be tangible. It's not like something you create a hypothesis which could you not even measure. then that doesn't really make sense. Okay, so you can make a hypothesis like I'm going to count all the stars in the sky. That's not possible. That's not measurable. That's not practically possible. Okay, I cannot. I can make a hypothesis like I want to measure all the neurons in my brain. That is possible, but then again, that is practically impossible. Theoretically possible, practically impossible. So always make hypotheses which are practically possible. That will make your lives easier. Okay, and that should be measurable. Okay, so once you have the hypothesis ready, then you have the pleasure of identifying what your objective is. Objective is a very specific statement that you are derived from the research question. For example, one example I have written it here: to evaluate the efficacy of the antibodies to lower the brain amyloid levels in a mouse model of AD. In this statement, I have already told that I am testing an antibody. I'm testing an antibody, and the parameter that I'm going to measure is lowering the brain amyloid levels. This means that I'm going to measure the amount of A beta protein inside my brain. So inside the brain of a mouse model. So I'm also specifying the model that I'm going to use. So you have started with the need, and then you have framed the research questions. From the research questions, you derived a hypothesis, and from the hypothesis, now you have derived a specific objective. So once you have objectives are ready, at this point of time, take a pause and look at your resources. What resources do you have in your laboratory? Do you have the resources to carry out ELISAs? Do you have the resources to carry out the biochemical assays? Do you have the resources to carry out RT-PCR? I mean, it's okay not to have resources. I mean, in private colleges, we do not have all those resources. We often have to collaborate. So one way what you could do is identify the key collaborators everywhere and try to talk to them about your research project. And then try to say, okay, you are very good in carrying out RT-PCR. Why don't you do carry out RT-PCR for this particular gene from my sample? I'm sure that no one would say no. Okay. So be very realistic in terms of what resources are available in your college or an institution. If they're not available, just collaborate. Okay, I mean, collaboration happens when there is a win-win situation. Okay, I am also benefited, you are also benefited. That's the way how you are going to do a collaboration. But then once you have the resources, you have everything set, then you have to choose a good animal model. Now, what is a good animal model? We have previously touched upon it. An animal model is a good animal model which is having a face validity. I mean, by looking at it, it should have a particular disease characteristic. Then it should have a construct validity. The reason why it is showing face validity is because the construct of that particular disease in humans is same as in the same animal model, right? So the construct is also same. These, if face validity and construct validity are true, then only whatever data that you're deriving from the animal, it should be applicable to the humans. So the predictive validity should be there. So that's what is a good animal model. Okay, and also you have to ensure that you have all the materials, all the tools, and all the equipment that is required for the experiment beforehand. It's not like you have bought all the animals, they are ready, and now you're looking after the materials. That's a very, very bad way to begin with. From A to Z, everything needs to be listed down and everything needs to be accounted for. And when there is a checkbox, I have a checkbox, checklist. If everything is there, then only go ahead and buy the animals or procure the animals. Okay. So once the animals are also ready, then then the critical step is designing your animal experiment. Now, what do you mean by design? Why do you have to design? I mean, animal experiments. One of the bad what do you call narrative of this animal experimentation is that people say that they're not reliable, nor they're reproducible. What do I really mean when they say animal experiments are not reliable? What I mean to say is that, for example, if you have to rely on the animal experiments, Alzheimer's diseases they were cured some 20 years back, but then that translation is not there. Meaning, the data that you are going to produce from the animals, it is not applicable to humans. Meaning, it's not reliable. So the animal model that you have used, it is not good. The same is the case of, let us take the cancer, right? Cancer was cured long back in animals, but then again in humans is still struggling. So reliability, if you properly design your experiment, the reliability of an animal experiment would be more. And then the another feature is called as a reproducibility. These two are the key features of your experiment. What is reproducibility? Reproducibility means if you are doing an experiment and you get a particular result, if XYZ after some time he does the same experiment, he should also get the same result. And often that doesn't happen. So someone finds does an experiment XYZ molecule cures coronavirus or COVID-19, and then someone tries to reproduce that experiment, he fails, meaning that animal experiment was not properly designed. That is why the reproducibility of that particular experiment is very, very low. And these two, reliability and poor reliability, I mean, no, almost no reliability and very poor reproducibility are the reasons why most of the people, they do not prefer to do animal experiments. I mean, if you don't have a proper animal model, then what is the point in even doing the animal experiment if you cannot translate it to humans? And if you are generating the data and if you cannot reproduce your own data, what is the point in even doing the animal experiment? So these are the two main issues that most of the people who are against the animal experiment, they always put up, that they are not reliable nor reproducible. So you have to design your animal experiment in such a way that it is more reliable and reproducible. At the same time, I am sure that your institution has a animal house, and this animal house has to bind to with CPCSA regulations, and you have to get your study protocol approved by your institutional animal ethics committee approved. Right? You have to submit your application to IAC committee. This committee will evaluate your study and then it will approve it. Then only you are allowed to do this animal experimentation. So, I mean, coming to this slide of an experimental design. I mean, the design is the entire experiment in one go. What is that you're going to do? And one best way of designing is always keep it simple. Don't try to answer too many research questions in one experiment, right? Previously, I told you that you have to frame research questions, and basing on the research questions, there will be hypothesis. Basing on the hypothesis, there will be objective. Basing on the objective, there will be an experiment being done. Keep it simple, meaning try to address one research question with one experiment. For example, there are 5 students in the M-Farm who will be doing their research project. All 5 can work on the same research question, very broad one, but then again, they can all address a different aspect of it. That way, the reliability, because the same person, the different person is doing a similar question, they're answering the similar research question, but then again, that person would be answering in a different way. So if you answer the same research question and using different methodologies, uska reliability बहुत ज़्यादा होगा. That reliability will be more and reproducibility will be more. So you have to keep it very simple, and simplicity is possible when you divide the research questions amongst yourselves. Okay, if there are 10 research students are there, divide it into 5. Everyone take care of 2 research questions, and when you combine it, it will be a beautiful study, because not everyone is focused on everything, but then everyone develops. For example, someone is very much good in animal behavior, so let that person pursue a research question that relates to the animal behavior. Some person is very good at doing ELISAs, so let that person pursue a research question which related to ELISA. So you divide the task. At the end, when you bring up the data from all these 5 different groups, a beautiful study would be there. And then in experimental design, as I already talked about, you have to choose the animal model, or do you want to choose a mouse model, or do you want to go for a rat model? Both of them have advantages and disadvantages. You have to discuss with your guide which animal model will be useful for you, and also you have to take whether you are going to use both males and females, or just using only one particular gender. But if at all there is any specificity for a particular gender, you better have a good explanation for it before, because IAEC committee is definitely going to ask for it. And then another thing comes here is experimental design and identify the experimental unit. What is your experimental unit? Experimental unit is the fundamental unit in your design that is actually generating the data. For example, if my experimental unit is a rat, then if I am deriving the data from a single rat, then my experimental unit is a single animal, right? Sometimes it might be a brain region, it might be a group of animals. So Experimental unit refers to the fundamental unit that is actually generating the data point. That's the experimental unit. Then how many groups it should have? As I said, keep it simple. Two groups to begin with. I mean, if you want to have a treatment, if it's a treatment design, so control versus treatment. One dose. I want to test I want to get a dose response. So maximum 3 doses or 2 doses. Keep it simple. जितना ज़्यादा कम groups होगा, उतना ही ज़्यादा reliable होगा. So you have to design your experiment in the most simplistic way, and also you have to be very much clear in why you're doing that particular excuse me, why are you using that particular animal model, and also why that specific experimental unit is being used. And then very important aspect of experimental design is randomization, blinding, and controls. Now you have to ensure that the treatments that you're giving, or the numbering that you're giving, they're almost random. Why randomization is important? Because you want to avoid bias. For example, one animal is very much active, so you don't want to give it a particular treatment, or you don't want to change your own behavior. Behavior of the experimenter should not be biased by the behavior of the animal. So randomization is very much important. How could you ensure that randomization is being done? You can blindly give random numbers to, I mean, identifying the numbers, identifying the cages. You can randomly ensure that, give them tag them with the numbers, or blinding them is also one way of ensuring randomization. So you don't know the drug that you're injecting, whether it is a controlled one or whether it is a treatment one. So you're blinding yourself with the dose or the treatment. And also having controls also important. So always controls play a very important role. It all depends upon the number of factors you're using. So if it is only treatment is the only difference between the that you want to evaluate, then there is one treatment or one control. So if there are two independent factors, then you will have to have two independent controls. So now the types of controls, the blinding and randomization, all these, they ensure that there is no systematic bias that is seeps into your experimental design. And if bias gets into your design, then probably that is your reliability of the experimental experiment is going to go down, and also reproducibility is going to be affected. Okay, so another important point that you want to talk about is about the sample size. Now, this is the most commonly asked question: how many animals should I use? I mean, I have worked in here in Indian labs where there is a limitation with respect to the resources. I totally agree, but then there is this magic number of six animals per group, and nowadays people are even going down even to 4 or 5. Where did this number come from? Where did this magic number come from? So it all came from some good man. He did a pilot study and found out in his design, in his experiment, in his method, the variation was minimal by using 6 animals, and that tradition is still continuing so far. Okay, so my advice to you is, before deciding for the sample size, just do a small pilot. Just do a small pilot in your own lab where you're going to measure, so that you know what is the variation. What is the variation? For example, all of you would be measuring something in your experiment, right? It could be your brain amyloid, it could be the behavior of the animal. So that variation needs to be quantified. What do you mean by variation? Standard deviation. Okay, there is a parameter, and no animal is going to produce the same data. So what is the variation? So if the variation is very high, most often what happens is, when you're doing your behavioral studies, the variation could be as high as 50 to 70%. Trust me, this is true. 70% of the differences you see between the groups is because of the variation you see in the testing itself. So if you come out with a treatment size, effect size of 20%, that doesn't make any sense. So always do your pilot study, identify the standard deviation kitna hai, and try to see reduce the variation, and based upon that use your sample size. For example, aapka standard deviation is very low, that you don't need to have a high sample size. If your standard deviation is very low as 1% or 2%, you don't need to have even 6 animals. However, For as in the case of behavioral studies, if your sample, if your variation is as high as 25 to 30%, then please have at least 10 animals per group, so that the reliability of the data that you are that you are producing, it is that it is reliable. Otherwise, I mean, it would be totally unreliable, or no one will be able to reproduce the data that you're doing, and it would be waste of resources and also waste of valuable animal life. So sample size depends upon the variation present in the animals, variation present in the methodology, variation the way you're doing, and then again that can be estimated using a pilot test. So standard deviations are very much important to know a priori. If someone has done, your seniors have done this experiment, try to get their data and see how big are those error bars. If there are error bars are very big, then go for a higher sample size. If error bars are very small, then probably you don't need to have that a big sample size. But then again, behavioral studies में error bars 1% है, तो don't trust that. Behavioral studies में 1% error bar हो ही नहीं सकते. It could be as big as 20%. It could be as big as 50%, because animals behave differently. They're animals. They're not disciplined to, or they don't know how to behave in a particular test, and that's natural, right? So don't expect variation from the in your data, and that variation needs to be quantified, and based upon that varied data you have to have your sample size. And then also you have to decide about the treatment, how many doses you're going to use, which route you're going to use, what should be the duration of the treatment. Again, the golden rule is always use the number of doses or the routes or the duration whichever is animal friendly. Now, why should it be animal friendly? It shouldn't be friendly to the hypothesis, it should be friendly to me. No, it is the animal that is producing you the data. And if the animal gets stressed because of your design, for example, your experimental design says that get the blood samples every other day. So basically will be puncturing the animal every other day. So that animal would be under enormous stress. Trust me, the data that you derive from a stressed animal, it is as good as a garbage. It won't be useful for anything. It will do no good. So always use animal friendly doses, animal friendly routes, and animal friendly duration. For example, I had to test some antibody, or I had to test a particular chemical. The most obvious way is give it by oral gavage, right? That's most often done. You have an extract, you take it, the animal, and you give the oral gavage. That itself is very stressed. I mean, would you agree if someone gives a gavage to you? No, you will be very much stressed. So why don't you think that animal should be stressed? And when stress axis of the animal gets kicks in, then that will totally what you call change the pathomechanism. So do not introduce stress to the animals, because the animal responds to the stress, and in response to the stress, it expresses genes which would actually interfere with the experiments. That's not what you want. So what do you really mean by animal friendly? If you want to give a particular chemical for a treatment, give it in its drinking water. Now animals, you may not be able to give a very high doses, but then you may be able to accumulate those doses in the drinking water over a period of time. For example, for one experiment, I was actually using monocrotophos, but then monocrotophos doses, the animals don't like to drink it. What I did was I reduced the dose, mixed it in the water, and gave it along for some duration of time. It did induce oxidative stress over a period of time. Okay, fine, my animals are fine. I'm not able to complete my experiment in 1 day or 2 days, or in 1 week. My experiment will prolong to a month. For 1 month, my animals will drink water and food, but at the same time, they're not stressed. They're still being exposed to the chemical. They're still being oxidative stress being induced, but then again, they are friendly. Now, how do I know that they are friendly? If you take an animal onto your palm, and if it is not escaping from your palm, then it is friendly, then it trusts you, right? Then it is not stressed. So there are different ways how you can, that's a totally a different 1 hour class that would take how to be animal friendly. And trust me, if your animal is cooperating with you, it's friendly with you, the error bars, the standard deviation, whatever you measure, it will be tighter than the animals that is very erratic, that is frightened. that is scared. If an animal tries to escape or if it pees on your palm, then basically it means that it is stressed and don't use that animal. I mean, if you use that animal, it will end up as a as an outlier, that data point. So trust me, you have to devise your experimental design in such a way that it puts minimal to minimal stress on the animal. And who would be very very happy because of this? Your IAAC committee will be very happy, right? Because the very purpose of CPCC regulation is to make sure that the animals are not harmed. And trust me, you can design experiments making sure that animals are not stressed. People are doing it. I have done it. You can also do it. So there is this checklist I have derived. This is called as a prepare checklist for designing an experiment. It has all the components related to the design of a study, from literature searches to the legal issues involved. How to create a, have you created a hypothesis, and have you checked enough of systematic review? You can use this checklist for yourself. Okay, I have done this hypothesis, so this is my hypothesis, and I have assessed its relevance. I have checked these databases. So I have identified successfully the knowledge gap. And then there are any legal issues involved in it. Now, you cannot do animal experimentations with embryonic stem cells. It's not allowed. There are certain regulations you have to follow, right? And also there are certain ethical issues. You have to get past this IAC guidelines. You have to get your study protocol approved by the IAC, and you also have to check whether these animals will be suffering from pain or not. If they are suffering from pain, what the things that you're going to do. So all these things have to be incorporated. They have to be checked even before you begin your experiment. Okay, and then again there is also a checklist for experimental design. So do you have any prior data, right? Because what is your statistical power? Statistical power means the number of animals that you're going to include. What would be your statistical significance of levels? What is your define your experimental? All these things you have to do it a priori. And these this prepare checklist, which is available from the prepare consortium, this checklist will enable you to see to it that all the different points are addressed. Okay, so again it also this also has got for example your study involves the personnel requiring some training, right? For example, if you have to derive the blood of the animal every week, so how are you going to get the blood sampling? Retroorbital nexus in निकालना अच्छी बात नहीं है. You have to get it from the saphenous vein. Are you well trained to derive blood from the saphenous vein? If not, you have to get trained even before you start your experiment. So though that kind of training and education, and what type of limitation your facility has, and what is your time scale, and what sort of funding you have, all these things have to be sat down, and they have to be discussed with your guide, and they have to be checked, so that you take a proper decision whether to move on with this study or not. Now why should you do it? Because if you do it for the sake of doing, then probably it would be spending some money, you will be spending some animal lives, but then it is no use to neither to you nor to the society. Okay, and that you don't want to do. So you have to check all these checklists and be sure that it is worthy, there is a benefit of doing this experiment, then only you have to proceed further. So apart from that, there is also a quality component in this checklist. Please go through it. For example, you're testing some test substance, how pure it is. Can you obtain it in pure form or not? And those kind of things you can, there is also related to the experimental animals. Are these experimental animals wild animals? Have they been backcrossed sufficiently? Is there is any issues with the supply? All those kind of things have to be decided a priori. When you do it a priori, not only that, what would you do if some animal dies in between? Would you perform a necropsy? What sort of things you have to do? All those things have to be checked even before you test it. And you have to make sure that all the protocols are in place. Should you want to have perform a necropsy, you just have to pick up that procedure and you do it, right? And you have to report it appropriately. And what would you do for example, if your experiment involves pain to the animal? What would be the limit wherein which you're going to at the point in which you're going to kill that particular mouse or rat, because you don't want to put to further any pain? So all those have to be discussed with your guide, discuss with your animal facility manager, and then you have to decide a priori. So all this ensures that very few animals would be affected, or animals would be in comfort, and animal ethics would be followed, and IAC guidelines will be followed, and also the reliability and the reproducibility of the experiment that you're carrying out, that would be good enough. So these are just some of the tips that I have. First one, he says that don't fall for p-values. Most of the time I see that students, they fall for p-values. Okay, so the p-values means they have read a research article, and the difference between the control and the this treatment group is the p-value is 0.0001. No, p-values don't tell anything about the experiment. P-values tell you only about one thing. What is that one thing p-value tells you? P-value tells you about the probability the control and treatments are similar is less than 0.0001. So, okay, fine, my control is not similar to treatment, but then what extra information does it give? It doesn't give any extra information. The extra information, the useful information is actually given with effect size. What is the difference in the mean values between the treatment and the control groups? That is what is more relevant to you. For example, if you are working in an experiment, the difference between the control and the treatment is more than 50%. Yeah, but then still its p-value is not significant. Fine, the difference is just by chance. But what about the difference between the control and the treatment is less than 10%, but then p-value is less than 0.001, meaning there is statistical significance, but there is no clinical significance for it. What do I mean? For example, I have a drug that reduces the blood pressure, right? If the drug is capable of reducing the blood pressure by 10 mm of Hg and the p-value is 0.001, would you mean that that would be clinically significant? No, clinically significant नहीं है. Clinically significance तब आएगा when there is a big effect size, right? And also try to work with effect sizes more than 50%. Why? Because always you do the experiment, the effect size is 50%. The other person does the experiment, the effect size falls 40%. So the other person does it is done in some company 30%. Always the effect size falls down. Always the first person, this is called as a beginner's curse. The first person to do the experiment, you always have a big difference between the treatment and the control. But eventually, when that same experiment is being done by other guys, the effect size decreases. So the most useful parameter for you is the effect size. If you're reading a research article, look at the effect size. Okay, don't pay too much attention about the p-values. Also look at the variation. If you are trying to look at the parameter which is a biochemical assay, see to it that the variation is below 2% or below 5%, then only trust that data. For example, if the person is doing a BC assay or a Bradford assay and the variation or standard deviation is more than 25%, don't trust that data. That person needs to optimize that particular protocol, bring it below 5%. For example, someone is testing the behavior and the percentage of variation or standard deviation is less than 15%, okay, that's reliable. But if the behavioral standard deviation itself is more than 40 to 50%, then probably that test is of no use, because even if the p-value is significant, the difference between the control or the clinical significance would be very much varying. Because अगर 50% variation से हो रहा है, so what is the contribution of your treatment? I mean, you can that is not a good study itself. And also, focus on the effect size and also keep the variation very low. And again, optimize your sample sizes in terms of animals. I mean, ask if necessary. It is okay to ask 10 to 15 and a number of animals if you think the standard deviation of your measurement is higher, because it is better to do one experiment rather than do 10 different experiments with small sample sizes. You will be saving more animals, you will be saving more resources. So optimize your sample size. Don't hesitate to ask your IAC committee members for more animals. But then again, the science behind it should be more. You have to show the power calculation that standard deviation is more. That's why I need more animals. Okay. And where do I get that confidence from? Always do a pilot study and get an idea about the standard deviation of the parameters that you're measuring. And also collaborate and network. I mean, if you don't have a particular methodology in your lab, just collaborate. I mean, in my lab, I have a particular assay. In your lab, you have a particular assay. We can both collaborate with each other in a beneficial way. Okay, you participate in my project, I participate in your project. I do your samples, you do my samples. So that way, we can both be partners and we can both get the publications and the output, whatever it is there. So this way, the research can progress. So these are some of the tips I have. I think I have already completed my 12:30 time, but I have not started the examples of some of the experiments. But then I don't want to push you into excess time. Iswar Garu, if there is more time, I will proceed. Otherwise, I would like to conclude here itself. You
Not a
can
problem, sir.
talk
Not a problem. You can proceed.
all right, okay, great. So I would like to have prepared two experiments to show you from my own studies, these animal experiments. The first experiment is all about the biochemical assays, and the second experiment is about the neurobehavioral tests. So let us see one by one. So even before going there, uh, let me actually show you tell you briefly about what is Alzheimer's disease. This is the experiment that we've done in Stockholm, Sweden. So these are the brain sections from post-mortem brain section from human brains. So the one you see here, they are from a control person who died for natural causes, and this is the cortical regions in the brain. And this is the brain section from a person who died of Alzheimer's disease. And these are the again the cortical sections. As you can see, the entire cortex is full of these toxic proteins called as beta amyloid protein, and these beta amyloid proteins are not at all present anywhere. They are one or two. At the same time, you can also find that in Alzheimer's disease patients there is this tau proteins are present in their brain. They are called as tangles, but they're totally absent in normal person. So post-mortem tissue, so this already tells you that this disease might be related to this particular protein, beta amyloid protein or tau tangles. So, but then again, is Alzheimer's disease caused by A beta? That I don't know. That's a knowledge gap. Okay. Is the memory loss in Alzheimer's disease related to the neurofibrillary tangles? That I don't know. That is a knowledge gap. So if you really look at the mechanism of the plaque, what you would see is that there is this protein present on the cell surface called as the APP protein, which is sequentially cleaved by these secretases, and that releases the beta amyloid peptide protein that is released into the extracellular space. This extracellular oligomers, they combine together to form a plaque. And these plaque, as you can see, it is very toxic. It kills the neurons surrounding by, resulting in the formation of the neurofibrillary tangle. So this particular mechanism, you can derive it even in the petri dish. But then again, is it really true? In order to ascertain that, you need to have an animal study. So if I want to say that, okay, I have a plaque in the extracellular brain region. So can I remove the plaque with an antibody? That's a research question that I can ask. Okay, so you have to study, you have to do a proper literature survey, and you have to derive your own research questions. You have to sit with your guide, and you have to sit with your friends. You have to sit and communicate with your collaborators and try to derive those research questions. And then you can design your experiment. And then once you have identified the knowledge gap, then is there any need for the, for example, in this study, what I want to show in this slide is that there is no treatment for Alzheimer's disease. All the drugs that were done during the past 10 to 15 years, all of them have failed. And then that already tells us the society needs a drug. So how about trying to cure Alzheimer's disease with an antibody? That we can do. So there is a need, there is a gap, and there is a need. So we can go ahead and plan our study. So this is a study that we have done. It is called we called it as a IVIG study, that is intravenous immunoglobulins. So this is a set of 4 different experiments, but then I will show you one experiment. I mean, I kept the design very simple, only 2 groups, the saline and the antibody group. The antibody are called IVIG, intravenous immunoglobulins. These are human intravenous immunoglobulins at a dose of 1 gram per kg, which is quite a high dose. And then you can see n is equal to 12 to 15. Now you might be wondering, why did I take 15 animals per each group? Am I very rich guy? No. The reason why I took 15 animals is because I would be measuring the memories of these animals using Morris water maze. और Morris water maze में standard deviation बहुत ज़्यादा होता है. Standard deviation is very much high. So in order to beat that standard deviation, we need to have a higher sample size. And that's how I could convince the IIC people, no, I need a 15 animals. They would always like to bring down the animal size. So sample size, but then you have to be very strict. No, if I reduce the animal size, the reliability and reproducibility of my study would be very, very less. Trust me, for this experiment, the same experiment was repeated in 4 different countries and the result was same. Had I been compromised with 6 animals or 5 animals, I might have got a different result, and actually that might have been a reproduction might have been different one, right? So I take pride in saying that at that point of time, I stood my ground and asked for 15 animals. I did this experiment. I kept the design very simple, only 2 groups, Saline and IVIG, and I asked for this particular model called as APP Swedish PS1 and DE9 transgenic mice, and I gave IP injections. Now you might be wondering, why did you give IP injections? These are antibodies. That's the only way. But then how do I make sure that the mice that I used, they are comfortable or they're friendly? What I used to do is every week I will dose the animal only once. Okay. So that's the reason why I give a very high dose. But then again, the animal might be stressed. No, when you give an animal once per week for 4 months, that animal is actually acclimatized to you. Even before I give the drug, what I used to do is every day I used to go to the animal room, take the mice into my hands, and put it in the same position, do nothing, and put it back. Over a week of time, it gets used to this. So whenever it sees me, it tries, okay, this guy is going to come, he is going to hold me. I have to just relax my entire abdomen so that he will do his thing and will go. And same thing happens. And over a period of some 1 month, I think all the animals will learn to cooperate with the experimenter so that you can go ahead and give your dose and come back. So here I have used IP, and trust me, there were no, it was so much nice that even after giving the injection, if I keep the mouse in my palm, it will not escape from my palm. Meaning it trusts me even after giving all those injections for 4 months. For 4 months, once per week, that would be close to 40 injections, right? So we have done that, and the compliance with the animals was great. It all depends upon how you behave with the animals. If you are polite, if you're gentle, then they will be gentle with you. They will cooperate with you. You do your thing and you leave. Okay. So the first question we asked was, does the antibodies cross the blood-brain barrier? That I tried to answer using this immunohistochemistry. And these are the brain sections. As you can see, all those antibodies, they made a reaction, and there was a gradient from the rostral side to the caudal side, indicating that there was the antibody inside the brain. And this is my control section, or the section from the animal that was treated just with the saline, and you can see that the hippocampus was totally devoid of any antibody. This gives me the evidence that yes, the antibody entered it. And then we employed this triple immunohistochemistry to find out where that antibody is. So antibody human IgG is stained with blue color, with the green color, and then the microglial cells and the APT were also stained using fluorescent antibodies. And then you see that all those were in the same microenvironment, indicating that my antibody crossed the blood-brain barrier, reached the amyloid plaques, it decorated the amyloid plaques, and those amyloid plaques are surrounded by the microglial cells. So the same animal study I used also to test whether these 3 mechanisms are true or false. Okay, one is microglia mediated mechanism, one is the direct dissolution mechanism, and another is peripheral sync mechanism. I don't want to go one by one, but then again, I derived data from ELISAs and found out, and also from immunochemistry, and found out that direct dissolution is not a mechanism. I also carried out the plasma A beta levels assessments and found out that peripheral sync is not a mechanism. I also carried out the microglia stainings, and what I found out was that there was no difference in the number of microglia, and also there was no difference in the number of CD68. These are the markers that can be used, and hence these mechanisms are not there. So I have used the same simple experiment and tested three different mechanisms. So that is the beauty of the in vivo studies. You can test different mechanisms. You just have to be smart enough to target all different mechanisms. Okay, so that can you can get it by discussing with your guide, sitting with him and deciding which way you have to produce. But then again, I mean that's also a point of disappointment for us that we have done so much of time, spent so much, we didn't see any differences. But then again, when we use this particular CD45 microglia marker, we saw a big difference between the saline treated and the antibody treated animals. That indicates that maybe the number of microglia is not decreasing, but the inflammation, the brain inflammation is being addressed. So I did a triple immunohistochemistry studies again to differentiate between the different phenotypes of the microglia, and I used EBA, CD45, both the double stainings, and what I found was yes, there was a treatment difference, and the treatment difference was seen in different types of microglial cells, right? So there were type A microglial cells, there was no difference in it, but then again there was a difference in type B and type C microglial cells. All this was possible by the same in vivo mechanism, in vivo study that we designed previously. So once I know that it's the inflammation, then I quickly went to the RT-PCR and asked the same question: is it reducing the cytokine level, gene expression? And yes, it was indeed reducing the TNF alpha levels. But then again, the effect was not seen with the IL-1 beta. So again, there was a relationship between the activation of microglia and the cytokine. All those kind of things you can carry out. And then since my inflammation was suppressed, I made a hypothesis कि अगर inflammation is less, then probably neurogenesis must be high, and that exactly what it was found. Because you treated with HIV-Ig, there were plenty of newborn neurons as indicated by the double cortin staining can be seen, and these are the saline treated ones, and these are the IVIG treated ones. So and then again, the same study was also used to find out whether there was any toxic reactions as well. So and we did not find any bleed, because typically when you give antibodies, there is this chances of bleeding inside the brain. So that we try to assess it, and we found that it's absolutely safe. And trust me, the same study was carried out in Canada, the same study was carried out in France, the same study was carried out in Belgium, and all of them have come up with the same kind of a output. So what I want to say is that if you plan your study properly, the reliability as well the reproducibility will be high. I think Iswar Garu, I think we have taken too much of our time. I do not have time to finish this inflammation. This another experiment where in which I tried to show the behavioral aspects. But then again, the point I want to make is that uh, in behavioral studies, the what do you call, the variation would be very high. Maybe I can spend time on this Morris water maze. So in this particular experiment, what we did was there were four different groups. There were two different genotypes were there. So what we did was we used to leave the animal in the tank, and that animal has to swim, and it has to find out a hidden platform, and a camera above it used to detect to find out its path. So the mouse which knows its path will directly go there, and the mouse which does not remember anything, it will go on. And this is the data that we got for the 5 days. As you can see, in the beginning of the trials, almost all the animals took somewhere between 50 to 20 seconds to reach the platform. The same animals, by the 5th day, all of them they take around just 10 seconds to reach the platform, meaning there is a learning, and this is a totally a valid experiment. Okay, however, you did not find the differences between the groups. That's a different thing. First thing you have to prove is that your method is working. Okay, so this is one, but then again, you can see the error bars here. The error bars are quite high, and this might be one of the reason why it did not work. Okay, I think I will, these are the final slide. The main thing that we checked today is that we learn how to find a knowledge gap, and we know that knowledge gap needs to be translated into a need for the study. From when there is a need and when there is a gap, then it's quite easy to frame a research question. That research question needs to be fragmented into smaller specific questions. From those specific questions, you get your hypothesis. Once your hypothesis is ready, you get your objectives, and once your objectives are ready, then you have to choose a very good animal model and design the experiment in such a way that it is simple as possible, less number of groups, and then less don't try to put too much of factors in it. For example, you're working with two different treatments. Don't do that. Simple. Keep it simple. Answer one research question at a time. Then the reliability, then the ease, or then and then interpreting the data would be much more easier. Make sure that you randomize, you blind yourself, you identify the sources of variation, follow all your methodologies meticulously, try to restrict your standard deviations to the acceptable levels. In case of biochemical assays, it should be less than 5%. In case of behavior, it should be less than 15%. And then only the data would be reliable, analyze it and report it. So these are the key things that I have covered in my lecture. I think I have already surpassed. I'm very sorry that I have taken too much of time. These are the acknowledgements to all my collaborators who have participated in these studies. And yeah, and these are some of the resources I have for you. Thank you very much for your attention. I would be very glad to take your questions. I know that I did not spend a lot of time with the experiments because we were already running short of this time. Hopefully, I will be able to spend time interacting with you. Thank you very much.
Thank you, sir. Thank you for the information. You have shared a lot of information regarding your research with us. And
Yeah.
personally, my question is, what are the methods to induce dementia which may result in Alzheimer's disease, and how can we evaluate in vitro, in the sense of in vitro, not in vivo, at the level of institution?
In vitro, for in vitro, sir, you need to have a cell biology lab, and then again you need to have a neuronal cell line or a microglial cell line. It is very difficult to have establish it. If you have already a well established cell line, that is fine. I have some of my friends who carry out in vitro. So in all my experiments I used to collaborate. I do not have the expertise with in vitro models, so we used to collaborate with Johanna. She is very much very expert in maintaining those cell lines. So but then again, you really need to ask the question, in vitro may to what extent you can, because most of the data that we got from in vitro experiment, they did not translate, because most of my in vitro experiments, they showed that this IVIG is going to work in humans, but then my animal studies, they showed that it is not going to work. So in vitro studies, they have their own limitations, because I mean, come on, whenever you're inducing Alzheimer-like pathology in vitro, you're using excessive amounts of A beta, probably in micromolar region, right? But then again, in animal studies and humans, micromolar region में नहीं होता है. It is in picomolar region. So the entire the context is different. So the reliability is very, very less to my. That is my opinion. Yeah. And if you want to induce Alzheimer like symptoms or pathology in animals, you can use transgenic animals. But if you really do not want to use transgenic animals, you can use oxidative stress inducers. They can stress the or neuronal stressors, for example monoprotophos, organophosphorus compounds. You can mix it in water and try to give them. Over a period of time, their neurons will die, and probably they might develop memory issues. I have not checked it, but then there are I have seen references to that. I hope I have answered your question.
Yes, yes, sir. To how much extent can Hellman's method be useful, sir, in order to evaluate the anticholinergic activity as well as DTNB reagent mostly?
To what extent? Animal studies.
Yes, yes. Hellman's method is there in order to evaluate stealth cholinesteroids.
Yeah, yeah.
To how much extent it will be useful in identifying the dementia as well as Alzheimer's disease?
See, the problem is with Hellman's thing, you can quantify or get some idea about the cholinergic pathway, but then again, is it related to the memory loss? The ultimate aim of using an animal model is to make sure that there is a memory loss first. The second, is this memory loss because of the mechanism that is relevant to human, or is it because I have given excessively the toxic doses of my monoprotophos, so much so that it shut down all the acetylcholinesterases, and then it put up a toxic reaction. I don't want toxic reaction. I want the mechanism that is replicative, or that is representative of what is happening in human condition. And as far as this animal model is concerned, that data I have not seen whether the fall in the cholinergic levels is it related to the memory loss or not. That link has not been established yet. If you are interested, you can pursue. Yes.
Right. Thank you, sir. Thank you so much for the information.
Yeah, if students want to communicate or ask any questions, I'll be happy to.
Right. The participants, if you have any queries, you can feel free to ask the speaker.
I think it's the same thing. Yeah, this is the lunch time, I know. I think there are there any questions?
If they don't have anything to speak,
Mhm. Okay,
let us conclude
good.
the session, sir.
Yeah.
I extend my thanks to our head of the department, Professor Narendra Babu sir, for giving such a free hand to organize this event. And as well as I thank our head of the institution, our principal, Professor Ramaran Nadendla sir, as well as management in order to organize this event in a great way. And primarily, I want to thank our today's speaker, Dr. Lakshman Sir Puli, who accepted our invitation immediately when we messaged in LinkedIn. And thank you so much, sir, for your great sharing of the information, as well as you shared as many things regarding how to plan the research, as well as how to proceed with animal experiments and all the things. And also the resources and references what you have gave to us in this presentation, they might make us confident to proceed in the way of experiment. Really, it is a very good design, experimental design what you have taught in this session. Thank you so much, and thanks to all the participants for your presence over here. And thank you so much. Let us conclude the session. You can leave the room. Thank you so much. Thank you so much, sir.
Yeah, thank you very much for this opportunity. And yeah, I mean, I thank your department head and as well as your principal, as well as your management. I'm always happy to share my knowledge. If at all any one of you have any questions, feel free to contact me. I'll be happy to discuss your experimental design, and we will also be happy to collaborate with you. I mean, we're also a private college, and I know that private colleges have limitations, so we can bring together our strengths and we can produce high quality research papers as well. So yeah, I will be looking forward for some collaborations from your college as well. And thank you very much. And yeah, have a nice
Thank you.
evening.
Thank you,
Thank
sir.
you.
Bye.
Bye bye.